In this topic, we're going to discuss the principles of immunity, antigens and antibodies. So we're going to look at what is an antibody and antigen, the features of an antibody, the structure of an antibody, the difference between self and non-self cells, the functions of antibodies, the classes of antibodies, and then I'm going to give you a little bit more information on monoclonal antibodies that are used in pregnancy test kits. Immunity is the ability of organisms to resist infection by protecting against disease-causing microorganisms that invade their bodies. So it involves the recognition of foreign material and the production of chemicals that help destroy it. So here you can see a pathogen, and on the surface of the pathogen are little proteins called antigens. So any molecule that the body recognizes as foreign, it's usually a protein, will initiate antibody formation. So this is called an antigen. The antibody, which is represented by the orange, is a protein called an immunoglobulin, which is made by B lymphocytes in response to these antigens. So the immune response is when these B lymphocytes respond to the presence of antigens by secreting antibodies. So let's have a look at antibodies in a little bit more detail. Antibodies are also known as immunoglobulins. They're proteins synthesized by cells in the blood by B lymphocytes. So when the body is invaded by foreign material, so an antigen, B lymphocytes produce antibodies that are going to react with the antigens on the surface of that pathogen. So antibodies are very specific to the antigen. As you can see on this antibody, you've got two sites at the end of the antibody where the antigens can bind. So antibodies comprise 4 polypeptide chains. You've got 2 heavy chains and 2 light chains. So notice how the heavy chain is in the middle and the light chains are those smaller chains on the outside there. Okay, let's label the different parts of the antibody. The yellow part at the top there represents the antigen binding sites. This means that the antibody can bind to 2 antigens at the same time. This is also called the variable region. The chains are held together by disulfide bridges, and they link the polypeptide chains and maintain the quaternary structure. The hinge region, represented by those dark blue circles, is flexible, so it gives flexibility when the antibody binds to the antigen. The constant region is the same in all antibodies, and it's where the antibody binds to a receptor on a macrophage. Now, to be able to defend the body from invasion by foreign material, B lymphocytes must be able to distinguish between the body's own cells, which you can see on the right, and chemicals on the left. So the body's own cells are called self cells, and foreign cells are called non-self cells. So here you can see a B lymphocyte travels around the body. It comes in contact with a self cell, recognizes it as its own from the own body, so it doesn't do anything. Then it comes into contact with a non-self cell and it binds to it, and then an immune response will be initiated. Now, if the B lymphocyte didn't recognize the body's own cells, it would produce antibodies that would destroy the organism's own tissues. So how do B lymphocytes recognize their own cells? So here you can see the B lymphocytes moving around the body, and they come into contact with self cells. They recognize them as their own body cells, so they leave them alone. Now, each antibody is produced by a different lymphocyte. In the fetus, the lymphocytes come into contact with their own cells. Infections in a fetus are quite rare because it's protected from the outside world by the placenta. So as I showed you earlier, the lymphocytes will collide with almost exclusively their own material, so self cells. Some lymphocytes do have receptors that will match the body's own cells. So you can see that represented by this light blue B cell. These lymphocytes will be selected against or destroyed, so that the only remaining lymphocytes are those that fit foreign material, so that the antibodies that are secreted will only be against foreign material and not against the body's own cells, the self cells. Let's have a look at the functions of the antibodies. The functions depend on the type of antigen to which the antibody binds. So the first is the antibodies can combine with viral or bacterial toxins and prevent the entry of these into host cells. They can attach to the bacterial flagella and immobilize the bacteria so that they are easier to engulf. They can cause agglutination of bacteria by attaching to multiple binding sites. They can work with other molecules to make holes in the bacterial cell wall so that water can enter into the bacterial cell, and because the bacterial cell wall is now weak, the bacterial cell will burst. They can coat the bacteria, providing recognition sites for phagocytes. Or they can act as antitoxins by combining with and neutralizing toxins. So here's another diagram to show you the different functions of the antibodies. So these include neutralization, agglutination of microbes, precipitation of dissolved antigens, and making holes in the bacterial cell wall. Right, let's move on to the different classes of antibodies. You've got IgG, which have got 2 binding sites. They're found in the blood and tissue fluid, and their function is to enhance the activity of macrophages, to act as antitoxins, and to cause agglutination. IgM have got 10 binding sites. So you can see that there are 5 antibodies, and each has got 2 binding sites, so it's 10 binding sites in total. They're found in the blood and tissue fluid, and they initiate the primary response, and they also cause agglutination. IgA have got 2 or 4 binding sites. They're found in your saliva, bronchial secretions, mucus secretions, and also in colostrum. So they inhibit bacteria binding to host cells or forming colonies on mucous membranes. And then lastly, you've got the IgE, which have got 2 binding sites. So they're found in your tissues, and they activate cells to make histamine. So they are involved in allergic responses to harmless compounds. So if you have hay fever, it's your IgE. Okay, a little bit of extra information on monoclonal antibodies. Now, when a pathogen invades the body, this pathogen has got many different antigens on its surface, and each antigen will induce a different B lymphocyte to multiply and clone itself. So you have a mixture of different antibodies being produced. In medicine, it's beneficial to produce a single type of antibody from just one clone of B lymphocytes, rather than having a mixture of antibodies. So these are known as monoclonal antibodies. Mono meaning one. So monoclonal antibody is an antibody produced by a single clone of cells or cell line, and it consists of identical antibody molecules. So where would we use monoclonal antibodies? Well, in a pregnancy testing kit. What you do is you've got immobilized antibodies on something called a urine dipstick. So this detects traces of a hormone called HCG, human chorionic gonadotrophin hormone, that gets released by the placenta. So if we look at the different stages, you can see that antibodies are tagged with a particle. So the antibodies are tagged with a particle, represented by those green drawings there. The HCG binds to the first antibody that's got a colored particle attached to it. This antigen-antibody complex then moves along the dipstick until it reaches the test zone, where it comes into contact with immobilized antibodies that are represented by the blue. These bind, and this is where you get your first line in the pregnancy test kit. The antibodies that aren't attached to the HCG then attach to the next zone of immobilized enzymes. This is the control zone and forms the second line in a pregnancy test kit.